Welcome to Tracking of the Beaten Track, an educational presentation in which I will provide an overview of the concepts, technical considerations, algorithms, and challenges of fiber tracking. I have no financial interests to disclose. First off, what is fiber tracking? Well, it's a technique that takes the local fiber orientation estimates that we can obtain from diffusion MRI scans, as you see here, and turns them into long-range connections. So if you want a definition of fiber tracking, I would say it is piecing together the local fiber orientations to infer the long-range connections in the brain. Although fiber tracking can be performed on non-brain tissues such as muscle, ligaments, and renal pyramids, its primary application domain has always been the central nervous system, and more particularly the brain's white matter. White matter is composed predominantly of bundles of myelinated nerve fibers, or axons, and these axons are long, thread-like extensions of nerve cells that connect specific brain regions and relay information by means of electrical impulses. Together, these fibers are tightly packed into fiber tracts, bundles or fasciculi, which are bundles of fibers that share a common origin and destination. At the highest level, we have what is called the connectome, or the full wiring diagram of the brain. Capturing this connectome can be considered the ultimate goal of fiber tracking, because it has the potential to provide unprecedented insight not only into the brain's architecture, but also into its function. Indeed, to understand how the brain operates as a whole, not only do we need to understand what cortical regions are active, it's also important that the physical connections that relay the information between these regions are mapped out. So how does tractography work? In its most straightforward form, called streamline tracking, we assume that each imaging voxel is characterized by a single predominant fiber orientation, and we piece together these local fiber orientations to obtain global trajectories. Mathematically, the set of local fiber orientations can be considered a vector field, and the global fiber trajectories are its streamlines. A streamline is any curve whose tangent is parallel to the vector field along its trajectory. So in order for a streamline to align with the vector field, its derivative has to match the vector field. So how do we solve this equation? This is a differential equation that can be solved by means of integration. The most intuitive way to perform this integration is by starting at a given point R0, called the seed point, obtaining the corresponding local fiber orientation V R0, called the step direction, and then following that direction for a short distance delta, called the step size, to obtain the next point R1 on the streamline. We can extend the streamline by repeating this procedure from the new point, and by performing this procedure iteratively, we can reconstruct the entire pathway. This integration scheme is called Euler integration or first order integration. Note that the vectors provided by diffusion MRI are merely orientations that carry no sense, so we have to track both along V and minus V to obtain the entire trajectory. And of course, we can track from as many seed points as we want. The individual trajectories that result from fiber tracking are called fiber tracks or pathways. An ensemble of tracks is sometimes called a tractogram. This can be a whole brain tractogram that covers the entire brain, as depicted here, but it can also be limited to a specific fiber bundle. At this point, it should be clear that fiber tracks are not to be confused with the actual white matter fibers. While the white matter fibers in our brain are physical objects, fiber tracks or streamlines obtained with fiber tracking are virtual entities that encompass no physical volume and that are related only indirectly to the nerve fibers. For the same reason, and for the reasons outlined in this presentation, the terms connectome and tractogram should not be interchanged freely without proper context. Let's now review the main technical considerations when designing a fiber tracking algorithm. One of the most defining and challenging aspects of designing a tracking algorithm is choosing the underlying model that relates the raw diffusion-weighted images to the local fiber orientations. All of these models rely on one fundamental assumption. When a number of nerve fibers align themselves along a common orientation, the diffusion of water molecules will be hindered to a greater extent across this orientation than along it. This enables us to figure out what the orientation of the underlying fibers is. It is important to realize that, in contrast to imaging techniques such as polarized light microscopy or electron microscopy, where individual fibers can be visualized directly, diffusion MRI can probe white matter orientations only indirectly by looking at the average diffusion pattern of the water molecules. A full review of the local fiber models underlying tractography is beyond the scope of this presentation, but I would like to briefly discuss the difference between using DTI versus a multi-fiber model in the context of fiber tracking. DTI is one of the most widely used models to obtain fiber orientation information, owing to its simplicity and its modest acquisition requirements. However, in voxels of complex fiber architecture, for example, crossing fibers or partial volume effect between adjacent bundles, the diffusion tensor is often a poor representation of the underlying fiber orientations. So in these examples, we have two fiber bundles interfacing at different angles. And while these two bundles can be made out clearly from the average diffusion pattern, the DTI model is just too simple and throws away this information. In the case of an acute angle, we can still obtain an average orientation, but in the case of a right angle, the resulting diffusion tensor no longer has a well-defined principal diffusion direction. So while DTI tractograms look very impressive even today, there is now a consensus that so-called higher-order methods that can accommodate multiple fiber orientations within a single voxel provide trajectories that are biologically much more plausible, and that DTI tracking should be deprecated. Typically, these high-order methods represent fiber orientations as a continuous function on the sphere, known as the fiber orientation density function, or fiber ODF, as you can see here on the right. Such a representation makes these models ideally suited to track through regions with complex fiber orientations. So if I switch back and forth between these two approaches, you can see that the difference is really night and day. So while these higher order methods can produce much better tractograms, one has to realize that the mapping from diffusion signal to fiber orientations is fundamentally ill-posed. This means that distinctly different fiber configurations such as bending, fanning, crossing, kissing can all give rise to the same MRI measurements, making it impossible to distinguish between these cases at the local voxel level. As a result, even the most advanced fiber tracking approaches will still be subject to modeling errors, and for this reason alone, fiber trackings should always be interpreted with caution. Once the fiber orientations are obtained for each voxel in the brain, the next challenge is linking them together. So we've already seen how this can be done using first order or Euler integration. An important drawback of Euler integration is that it assumes that the fiber orientation is constant at the length scale of the step size, which makes it highly susceptible to overshoots in curved bundles. If we move to higher order integration schemes, which take into account the variations of fiber orientation between the successive steps, these large integration errors can be vastly reduced. This image also nicely illustrates that the local step-by-step nature of fiber tracking makes it extremely susceptible to error propagation. Even small local errors, be it due to modeling errors, integration errors, or just noise, can accumulate along the way, causing tracts to veer off course completely. So where do we start the fiber tracking? In general, fiber tracking is performed by starting from a number of seed points that define a specific region of interest, or ROI. Often, these ROIs are defined by the user, relying on expert anatomical knowledge. Alternatively, they can be defined from atlas labels, or from cortical activation maps obtained with fMRI. An alternative to region of interest-based tracking is the use of whole brain tractography. Whole brain tractography can be achieved either by seeding from all white matter voxels or by solely seeding from those white matter voxels that are close to the gray matter interface, as this is where the fibers are known to originate from. A final aspect of streamline tracking is deciding when to stop. Two criteria are commonly used: local fiber orientation probability and local curvature. In the case of DTI tracking, it is common to stop a streamline when the fractional anisotropy falls below a certain threshold, as regions of low FA tend to be associated with high uncertainty in the principal diffusion direction, and therefore a large potential error for the next step. For higher order tracking, the obvious stopping criterion is the local fiber density along the current tracking orientation, allowing tracking even through regions of complex fiber architecture. The curvature threshold imposes a maximum local curvature of the tracks. If the angle between two successive steps is above a predefined threshold, the track is terminated. The idea is that any sudden change in trajectory is caused by artifacts, rather than biological reality. However, there are situations where this assumption of fiber continuity might be invalid. For example, Meyer's loop is a highly curved portion of the optic radiation, depicted here in green. When seeding at the LGN area, most fiber tracking approaches will favor straight trajectories and instead track exclusively towards the temporal pole, missing Meyer's loop almost entirely. Apart from this termination criteria, fiber tracking also often employs acceptance-rejection criteria. The most widely used criterion is that of minimal track length, rejecting short tracks on the assumption that they are spurious. More advanced criteria are based on anatomical priors and reject tracks that terminate within the white matter or cerebrospinal fluid and only accept tracks that connect different gray matter regions. So here we have a conventional whole brain tractogram that looks great on the surface. But if we inspect the endpoints of the fiber tracks, it shows a significant number of fiber tracks that are terminating prematurely within the white matter. For example, in regions of complex fiber architecture or at the interface with the CSF. Once we impose anatomical constraints, resulting fiber trajectories no longer stop prematurely within the white matter, but very evenly cover the white matter-gray matter interface as expected from anatomy. So if we have ticked all of the technical boxes, we've used a state-of-the-art local model that can capture multiple fiber orientations. We've designed an integration scheme that avoids overshoot. We've carefully seeded throughout the brain, and we have tweaked the propagation and termination criteria. Let's have a look at the kind of tractograms that we can get. So here's a slab visualization of a whole brain tractogram. You can easily make out all the major bundles as we scroll through the brain. If you look closely, you can even make out fiber crossings, for example, those of the corpus callosum and the corona radiata. We can also look at these in 3D. Here we have a rotating tractogram inside a translucent cortical surface. And you can see that with modern techniques we can get quite nice coverage of almost the entire cortical gray matter. Beautiful as these whole brain tractograms are, people will often want to look at just one specific bundle at a time. This is where track selection comes in. Typically, track selection is performed by defining regions of interest through which the bundle is known to pass. Tracks that don't pass through these regions are discarded, keeping only the bundle of interest. This technique has been successfully used to isolate many different fiber bundles, and as such it is sometimes referred to as virtual dissection. Virtual dissection is a very powerful segmentation tool for white matter bundles, and resulting segmentations are nowadays routinely used to study quantitative metrics, either from diffusion MRI or from other modalities, in specific fiber bundles only. For now, we have looked at the generic technical aspects of fiber tracking, but we have not touched upon the many different fiber tracking algorithms that are out there. It is beyond the scope of this presentation to review every published technique, but I will try to give a brief overview of families of tracking algorithms based on their most distinguishing features. A first distinction is that between deterministic and probabilistic approaches. Deterministic approaches produce the exact same trajectory each time they are run, and typically they follow the dominant or most likely pathway. Probabilistic approaches, on the other hand, throw in a random or stochastic element, where a different trajectory is explored on repeated runs. This allows to also explore non-dominant or less likely fiber trajectories. And instead of a single trajectory, they provide a distribution of trajectories. It is important to understand that there are basically two fundamentally different schools of probabilistic fiber tracking. The first school assumes that a limited number of dominant fiber orientations are present in each voxel. These local fiber orientations are subject to noise and modeling errors, resulting in a distribution of possible fiber orientations, also known as the uncertainty ODF or UODF. By drawing samples from the UODF, a distribution of fiber tracks can be generated that reflects this uncertainty. As a result, UODF tracking provides an indication of how precise fiber tracking results are, with more reproducible trajectories exhibiting higher visitation counts. Notable examples are FSL's Bayesian tracking or tracking based on bootstrapping. The second school assumes that each voxel contains a continuous distribution of fiber orientations, also known as the fiber ODF or FODF, and ignores the effects of noise and modeling errors. So by drawing random samples from the FODF, this approach is trying to capture the anatomical dispersion of the fibers. A notable example of this approach is Matrix's probabilistic FODF tracking. So here we have an example of probabilistic fiber tracking in the SLF using both approaches. On top we have probabilistic tracking by taking random samples from the uncertainty ODF obtained using the residual bootstrap, and below we have probabilistic tracking by taking random samples from the fiber ODF. While there is overlap in the resulting visitation count maps, it's clear that they both convey different and complementary information. The UODF approach tells you how reproducible tracking is, which for example depends a lot on the SNR of your data. The FODF tracking, on the other hand, tries to capture the physical destinations of the fibers. A second distinction that we can make is that between local and global approaches. In a local approach, we start from the acquired data. We then extract local fiber orientations using an appropriate model. And finally, we perform a localized, stepwise integration of these orientations. So at no point in this pipeline is there any feedback from the reconstructed fiber tracks back to the local fiber orientations or to the diffusion data. Local methods are fast and widely available, but they have important drawbacks. The most apparent one is probably that minor errors in the local fiber orientations can accumulate and significantly affect the final result. Another, lesser known downside is that local tractograms are typically a very poor predictor of the measured diffusion data, resulting in fiber tractograms that have very little quantitative meaning. Global methods, on the other hand, follow a completely different approach. They reconstruct all tracks simultaneously by finding the configuration that best describes the measured diffusion data. So we start with an initial whole brain tractogram, and from this we predict the measured diffusion data and compare it to the acquired data. Based on this residual, we then update the whole brain tractogram by adding, moving, and linking fiber segments. The whole brain tractogram is then iteratively refined in order to find the best match of the predicted and the acquired diffusion data. Because the way they are constructed, global approaches are much less susceptible to error accumulation, and the resulting tractograms predict the measured data by definition, making them much more quantitative. The main drawback of global tracking is that it has to rely on complex stochastic optimization procedures in order to find the optimal tractogram, making them much slower than local methods and less commonly implemented. Closely related to global tracking is the concept of tractogram filtering. While global fiber tracking uses a bottom-up approach, where a tractogram is directly constructed in one global optimization routine, tractogram filtering adopts a top-down approach, starting from a dense whole-brain tractogram that was obtained using a local tracking technique, and subsequently removing or reweighting tracts in order to match the local tract densities to the measured diffusion MRI data. So in a way, tractogram filtering is an approach that makes non-quantitative local tracking quantitative again. Okay, so now with all of the technicalities and the different fiber tracking approaches out of the way, it is now time to look at how we can extract meaningful numbers from these tractograms. The two most commonly attempted methods of quantification are voxel-wise track counts and point-to-point connection strengths. The idea of voxel-wise track counts, also known as track density imaging, is to create a dense whole-brain tractogram and then simply count the number of trajectories passing through each imaging voxel. It is then extremely tempting to use the resulting track densities as a surrogate for fiber density. The idea of point-to-point connection strengths is again to create a dense whole-brain tractogram and then count the number of trajectories connecting specific regions. Here, the number of trajectories is used as a surrogate measure for structural connectivity. Especially, these connection strengths are becoming increasingly popular as they are central in the field of connectomics. But can fiber tracking really deliver reliable quantification? I would like to argue that quantification of fiber tracking is really a slippery slope. A first issue with quantification is that fiber tracking tends to favor bundles that are short and simple over those that are long and complex. This is demonstrated clearly by this nifty simulation by Derek Jones. So he performed probabilistic fiber tracking from a single seed point along trajectories of varying lengths and complexities, but with essentially the same microstructural properties. And then he looked at visitation counts along each trajectory. If you look at the bundles targeting regions C and D, they should exhibit the exact same connection strength. However, because the bundle targeting C is much longer, the chances of encountering local errors as well as the extent of error accumulation increases, making it more likely for trajectories to terminate prematurely. The same thing happens in curving trajectories, or trajectories that encounter regions of complex fiber configuration. So fiber tracking is really favoring simple and short trajectories. What can we do about this? We can use anatomical priors to prevent premature termination. For example, priors that prevent stopping midway the white matter or inside the CSF. We can use global instead of local tracking to reduce the extent of error accumulation. And finally, we can adopt track filtering or global approaches to enforce fidelity of the tractogram to the diffusion data, which will avoid the artificially reduced densities in difficult to track regions. A second issue that plagues quantification of fiber tracking is that it tends to favor large bundles. So here I'm showing you voxel-wise track densities obtained from a whole brain tractogram. For reference, I'm also showing fiber densities directly obtained from local modeling, without the use of any tracking. You can immediately see that large and thick bundles like the corpus callosum exhibit an inflated number of fiber tracks, while the fibers close to the cortex are very much underdefined. So where does this bias come from? We know that it has to originate from the fiber tracking itself, because it was not present in the local fiber densities that the tracking was based on. So one of the main issues that causes this is the mechanism of whole brain seeding. It's easy to see that when you distribute seed points uniformly throughout the brain, large and thick bundles will be sampled by a much larger number of seed points, artificially boosting their local track counts. So how can we solve this issue? The fundamental issue here is that standard fiber tracking approaches have no mechanism in place that will ensure that the virtual track counts that we get from fiber tracking are consistent with the actual densities of the fibers encoded in the diffusion MRI signal. Such consistency can be established either by using a global tracking approach, which attains this by its very definition, or by applying a track filtering approach, like we did here. So for the whole brain tractogram to have any true quantitative meaning, it's really crucial that we either perform track filtering or adopt a global approach. What this slide also shows is that as far as quantifying fiber density, you're much better off extracting it directly from the local model. Indeed, why would you go through all the trouble of approaching something that you could obtain directly from the diffusion data in the first place? And then the final and probably most fundamental pitfall is that fiber tracking is really an ill-posed problem. We've already seen earlier in this presentation that locally many different fiber configurations can lead to the exact same voxel-wise diffusion signal. This also means that globally many different tractograms can explain the diffusion signal equally well, especially if we are dealing with so-called bottlenecks, which are regions shared by multiple fiber bundles. So here we have 4 different bundle configurations, which will almost be impossible to distinguish from each other using fiber tracking. The ramifications of this have been clearly demonstrated in the 2015 ISMRM Tractography Challenge. In this challenge, a synthetic diffusion-weighted dataset was generated based on a collection of synthetic fiber bundles that resembled those found in real brains, and expert teams from around the world were asked to provide their best whole brain tractograms. And this study came up with some damning results concerning the quantitative nature of fiber tracking. They found that on average, for every valid bundle reported by fiber tracking, more than 4 artificial bundles were tracked. What is alarming is that these artificial bundles approximate true anatomical structures very well. They connect cortical areas and they appear to be dense and very well structured. Even when tracking using the ground truth fiber orientations that were used to create the phantom, the number of false connections did not really go down. When looking at the cause of these valid bundles, most of them were associated with bottleneck regions, where tracking is fundamentally ill-posed. So I would like to argue that these results clearly support the inconvenient truth that current fiber tracking techniques cannot be used to reliably quantify long-range structural connectivity. So what do we need to do to reduce this ill-posed nature of fiber tracking? That is of course the million-dollar question, and it is still a topic that is actively being researched. In general, what we need to do is inject new information into our algorithms that goes beyond just the local fiber orientations. This can be non-local orientation information, for example by enforcing spatial continuity, as is already the case in global tracking approaches. It can be microstructural information, for example by tracking fiber populations with similar axon diameter or other microstructural properties. It can take the form of better anatomical priors learned from histology and anatomy, or maybe even priors tuned for specific fiber bundles. And finally, it can mean that we will have to resort to machine learning and AI and learn from many examples in order to overcome this challenge. So to conclude, I have shown that fiber tracking is a very complicated endeavor. This means that as a developer, you have to pay close attention to all the details, but also as a user, you really need to understand its inner workings and limitations very well. I have also shown that fiber tracking can deliver truly impressive qualitative delineations of known white matter structures. This enables bundle-specific region of interest analysis, and it can also be extremely useful to guide complicated neurosurgery. And finally, I hope that I've made it clear that quantification of fiber tracking is extremely difficult to do reliably. Even using the state-of-the-art techniques, tractograms will still contain many false positives and many false negatives, to the point that we can seriously start to question their quantitative meaning. So any attempt at quantification, especially quantification of long-range connections, should be approached with extreme caution. So that concludes my presentation. I hope you've enjoyed it. If you have questions or comments, feel free to email me or contact me on Twitter. Thank you for watching, and I hope to see many of you at the live Q&A session on diffusion, micro and macro on Sunday, the 9th of August.